Good evening to you once again, and welcome to SCH 214 Intermediary Metabolism and Biomembranes. Today, we're continuing to look at some industrial applications of enzymes so that you don't think that what we have been learning so far with the enzymes from last semester to this semester, we did some fundamentals of enzymology and looked at the actions of enzymes in metabolic processes. But this time we are going to take a look at how this knowledge that we have acquired is actually applied in industry in different fields. So that's what we're going to do today for you to appreciate the fact that science is very practical and not any abstract thing. So we're taking a look at the overview of industrial applications of enzymes, and you realize that enzymes are applied in various industries, very, very varied industries like the food and beverage industry. You have the application of enzymes there. In the textile industry, you also find applications of enzymes. The detergent industry, environmental applications, in the pulp and paper industry, you'll find applications of enzymes. Biofuel industry, you'll find applications of enzymes there. And I think pharmaceutical industry, we very much covered a lot of applications of enzymes there. And in the last lecture, we did look at how enzyme inhibition and kinetics is applied in drug discovery, drug manufacturing, and how some specific enzymes are targeted in some health conditions. So for pharmaceutical industry, I think we very much looked at how enzymes are applied in that field. I think the only other fields that we have not looked at very much will be the rest. Today we can't cover everything, like look at the examples of applications of enzymes and everything in a period of 2 hours. But what we'll do is we'll take some very interesting examples of the applications of these enzymes in various industries and look at them over this period. After that, it's a way of getting you introduced to the subject of applications of enzymes in industry so that if you have interest in it, you go, you read more, you research more, you see how enzymes are being used to do different things in different industries. And think even when you continue to do BTech top up, those who be interested in doing industrial biochemistry, there's some aspect of applications of enzymes there too that you cover more of these things there. So we'll continue and take a look at a few examples over the period. And by the time we finish, I think you have some interesting knowledge of applications of these enzymes, which will interest you to go and find out more about these applications. Most probably, the industry in which you have very varied, different applications of enzymes is the food and beverage industry. Perhaps because there are lots of, different kinds of food, food products, much more than maybe all the other areas. Maybe the only other area that enzymes have been applied in that can compete with the food industry will be the pharmaceutical industry. So you see that in the food industry, you have applications of enzymes in, say, in the production of dairy products, right? Milk products, different kinds of milk products, the milk themselves. You find there are different kinds of milks, say either to do with the amount of fat which is in the oil. So in some places you have milk which is having like different percentages of fats. You have maybe 10%, 50%, 60%, 40% fat, depending on your dietary habits. You choose one of these milks, or some of them almost 0% of these fats and oils in them. Or other milk products like maybe yogurt, and even yogurt. There are different grades of these. Cheese, you'll find application of enzymes in the production of these. In a bakery also, you'll find applications of different types of enzymes, particularly those that act on carbohydrates, the polysaccharides. In the beverage industry, also, either it is some fruit juices, or in the brewery, where the brewing of beer, there too you find applications of enzymes. Then of course, with some vegetables, like the smoothies of fruits and vegetables, also you find application of enzymes in all these fields. So there are so many kinds of foods. Depending on the jurisdiction in which you are, you meet different kinds of things, and many of these things have applications of enzymes in them. Now we continue to look at specific examples of the applications of these enzymes, taking maybe one industry after the other, and then we see how enzymes are applied in different ways. So one of the common applications of enzymes you come across is in the production of cheese. And you find the enzyme chymosin, which usually you find on the market being sold as rennet. Some of these products that you buy on the market, which is being used in the production of cheese, rennet, contains not only chymosin, but some other enzymes in addition to chymosin, like maybe rennin will also be there. Now, historically, the production of cheese locally had been done with the addition of extracts from the stomach of ruminants, like say some cows, that they take some extract from their stomach. They are ruminants, and so they have I think one of the stomachs, the abosom, they have the production of this enzyme, chymosin or rennin, which is responsible for the coagulation of milk. And this is historically you have the extraction from this. Of course, now there is production of this enzyme from different sources, where some of it is by genetic engineering, where you have the extract from the animal, the genetic code, which is responsible for coding for the protein of chymosin, is then recombined, with the process of recombination, so that you have a recombinant DNA of bacteria, which is then infused into the bacteria. Or sometimes you have yeast being used, so that the yeast begins to produce more of these enzymes, and then you have the yeast, after you have the production of the enzymes from it, you have it separated, and you can add it to milk, And when you add it to milk, it will cause coagulation of the milk. And you can then take the thick curds of the milk. The fluid part which is left is the whey. And then you have that solid cheese which is produced. Well, what you see there, I mean, this is in an industrial setting where you have cheese being produced. But if we want to look at it much more closely, to understand how such coagulation occurs, then we have to go a bit more into the chemistry of it, which is to say that the protein, there's a major protein in milk, which is casein. So casein is that major protein in milk. Now, what you see is that the casein is hydrophobic, which is to say it's not soluble in aqueous environment. But milk is aqueous. And so what you actually find is that casein forms such a homogeneous colloid with that aqueous environment, so that there is that homogeneous mixture that you don't find that the particles of milk settle at the bottom. No, normal milk will always be such a colloid. And so you have the structure of these, what you refer to as micelles. And these micelles, you have some calcium with some, phosphates. So you have the calcium phosphates, which are the black dots, and you have these submicelle. You have these micelles, the green one, and then you have submicelles with casein, the protein, which is bound on them. So you have such a cluster. It's like you have these particles which are homogeneously mixed with the aqueous part of the milk, and you don't find that this settles at any time. And so what it means is that when you introduce chymosin is a protease that goes to break down casein, which is red. So you have some of these micelles which have casein on them. That is the external one. Of course, if you go and look at the chemical structure of casein, you see it contains a lot of the amino acid proline. Then when you go back to look at the structure of the amino acid proline, you realize it has this internal, intramolecular cyclic structure, which makes proline hydrophobic at that side of the side chain. But of course, all amino acids have carboxylic acid group, and the carboxylic acid group is hydrophilic. So what it means is that the casein, which is bound onto these external micelles, makes it to have such interaction with the water molecules that are in milk, because the carboxylic acid groups on the casein can easily interact with water molecules, whilst you have that hydrophobic side binding onto these submicelles. But when you do have the enzyme chymosin introduced into milk, what chymosin does is that it goes to break down the casein. So the casein is a protein. So once it breaks down the casein, it means that the external part of the those casein those casein, aggregates that we just saw can then, bind together. So then you have, so if you see what you are saying here is that you have a casein micelles. And so you have, the kappa casein submicelles in them, the calcium phosphate, what we just saw. And when you introduce the enzyme, which is chymosin, of course, calcium there, acting, the calcium ions acting as a cofactor, you begin to have thick curds forming. And when such curd, such aggregates of casein are being produced, they will begin to settle. So then you have these curds of the milk settling at the bottom, and you have the liquid part at the top of it, which is what you refer to as the whey. So you can separate that solid part which has settled down, which is this casein aggregates that have settled, and that solid can then be processed into cheese. So you do have, apart from the chymosin that is being used for the production of cheese, you also do have other enzymes that are also used. I mean, nowadays you have production of different enzymes that are used for the same purpose, primarily because of preferences of different people. Some can be due to religious reasons, some dietary reasons. There are some people who are vegetarians and will not want to have, well, not even though the milk itself is coming from an animal source, but of course we do have even some cheese that are coming from some plant source. Some people would like to have the cheese produced by not animal enzymes, but by plant enzymes. And some too, maybe for religious reasons also, we want to have the enzymes being produced from some specific animals, but not others. And so you have different kinds of enzymes that are used, these days for the same purpose. And these enzymes, do have the milk clotting activity, what is intended to break down the casein micelles and for the milk to settle as some curds, which can then be separated for the production of the cheese. But then you can also have some residual activity of some of these enzymes, which can result into the breakdown of this casein protein into shorter peptides, which can result into some bitter taste in the milk. And so you have in the product the cheese, and so you do have some measurements for I mean, it's comparative to chymosin, because chymosin is taken as a standard that produces let's say the standard cheese. And so these other products which are used in industry are compared against chymosin to see the milk clotting activity as a ratio to, say, the proteolytic activity. And the lesser the proteolytic activity, the better, because then you will not be having some off taste, unintended taste, like the bitter taste that will be occurring in the cheese. So that's with cheese, that an enzyme or different enzymes can be used to cause the coagulation of the milk into a solid, which can be separated and then used for the production of different types of cheese. Now, you also will have other enzymes that are also used in the dairy industry, apart from cheese, which we have looked at much more. You have lactase. Lactase is an enzyme which we talked about to be an absolute specific enzyme which acts on lactose, breaking it down into glucose and galactose. Now, with that in mind, lactose is used in the production of, say, lactose-free milk, so that persons with lactose intolerance can consume such milks. Because, of course, once you have lactose, the milk sugar lactose broken down in such a product, it means for someone who is lactose intolerant, if the person consumes such a product, there will not be any lactose in that milk that can cause any adverse effects for such a person. I mean, what you see below is just a mechanism of the breakdown of lactose by lactase. I think we did look at it when we were looking at mechanism of action of enzymes, and this is a representation of such mechanisms. I just wanted to pay attention to because we did talk about, say, active sites of enzymes. You can see the particular amino acids that act in the active site of the enzyme. You see you have 2 aspartic acid residues at the active center. One is at 170th position, and so you see ASP 170, and the other aspartic acid is at the 231st position. So you have the ASP 231, and you see that the aspartic acid at 170th position, first of all, has some nucleophilic attack on that glycosidic bond between glucose and the other side of that can be some other, maybe glucose, whilst you have what is being shown, the monosaccharide that is being shown fully as galactose. And then you have the aspartic acid that is at the 231st position, also acting as an acid, acting on water molecule, deprotonating it, and then causing the hydroxyl group from the water to bind onto the position where you have the side group of that galactose molecule being removed from, so that now you have a full galactose molecule at the end. And then, of course, the other chain, if it's glucose, yes, glucose will be there. This is just showing galactose, which is broken off, so that if there's a different sugar, of course, if you take the lactose molecule, is glucose and galactose, so obviously the R means that is going to be glucose. And so you have the galactose is free, and you have the glucose molecule as well. So that is lactase being applied in that sense to produce special milks for lactose intolerant individuals. We did also see this reaction when we were looking at metabolism of lipids, where we looked at the breakdown of triacylglycerols into free fatty acids and glycerol. That's a hydrolytic reaction. And we did see lipases as enzymes that catalyze the hydrolysis of lipids. So the milk and fats, you have lipase, hydrolyzing milk fat. So milk contains some fat, and you have lipase that can break down these milk fats. And these two is to enhance flavor in cheese. So that is also in the production of cheese that you have some lipases being used. Apart from the chymosin, which we just looked at, you also have lipases which are also used in the production of cheese. Now we have Also, yes, lysozyme. Lysozyme was one of these enzymes that we did look at the uh, mechanism briefly, its mechanism of breaking down bacterial cell walls. And so we knew it to be a bacteriocytic enzyme because it will cause the cell death of the bacteria. So if such an enzyme is able to break down the bacterial cell walls and it is introduced into a milk product like the cheese, it will prevent the growth of, say, harmful bacteria. Now, in the production of cheese, if you do have some of these bacteria uh, that contaminates the product, what does happen is that you have what is referred to as late blowing in cheese. That is, these bacteria feed on the cheese, and they begin to produce the byproduct carbon dioxide, so that the cheese has bloats and has a lot of holes in it. Such cheese is not of so much of good quality. Good quality cheese will be more compact. Even if there are any holes, they are very, very tiny, not big ones. But if you do have some of these bacteria present in cheese, then you have what is being referred to as late blowing, and you have these large bubbles which are formed, and the cheese is full of big hollows in it. Niacin is another one, which also is used as a natural preservative. So you see, instead of using chemical preservatives, you have the addition of these enzymes that will extend the shelf life of the product. So because, for instance, niacin inhibits bacteria which cause spoilage of food. We did see that bacteria, for instance, causes food spoilage. Even fungi causes food spoilage due to the fact that these microorganisms feed on the food product and release their byproducts, which are toxin. And these toxins then cause food poisoning because they turn the food in, they make the food unwholesome, so that if a human you consume such a food that bacteria has fed on and has released these toxins into it, then its consumption can be harmful. So this is how you can also use the enzymes for as some natural preservatives instead of using chemical preservatives. Now in the brewery industry, we move on to the brewery industry, where you have amylases that act on carbohydrates. So in a brewery, what is done? In a brewery, carbohydrates are taken, from different sources. In some, these carbohydrates are coming from cereals. In Ghana, we have production of some beers from cassava, for instance. So, I mean, it means the source of the carbohydrates from which the alcohol is going to be produced, that is cassava. But much of production of beer is from cereals. And what is the scientific basis for that is the breakdown of these, the starches, which is a monosaccharide, the polysaccharides in these cereals are broken down by amylase. Just like, how we have salivary amylase, and salivary amylase, breaks down, starch into simple sugars. Now the amylase is introduced into the say the source of starch, which is in the cereals, so that the amylase then acts on the starch. Of course, the condition which is created for the activity of the amylase on the starch is some anaerobic condition, so that ethanol is produced. Because now you know from our metabolic processes, if the condition is aerobic, alcohol is not going to be produced. What you have being produced is acetyl-CoA, and then further that breaks down into carbon dioxide, and so you won't have the production of the ethanol. And so you do have alpha amylase that breaks down the the alpha 1,4 glycosidic bonds. And you also remember that we refer to the beta amylase, which is specific to breaking down the beta 1,4 glycosidic bonds. And the beta 1,4 glycosidic bonds, you find it in the indigestible part of carbohydrates. So you can imagine if you make use of only alpha amylase in the production of beer, for instance, so in the brewery, then what is going to happen is that it is only the alpha 1,4 glycosidic bonds that are going to get broken down. And so the indigestible carbohydrates which are present, having the beta amylase, will go to waste. So because of that, you have the combination of the alpha and the beta, and this then helps to increase the yield of production, because then such a mixture of having alpha and a beta amylase together will break down both alpha and beta 1,4 glycosidic bonds, and so you have more simple sugars which can then undergo the process of fermentation. And so then the microorganism yeast, which is introduced in this in the industrial bioprocessor, can then break down these simple sugars into alcohol by that process of fermentation. Now, again, in such a mixture, which is the enzyme mixture which is being introduced into the bioreactors, you also will remember that we did look at even if not last semester, I think in first year, when we were looking at some carbohydrates, we did talk about dextrins. And in dextrins, you have this branched chain polysaccharide, where you have the 1,4 glycosidic bonds in addition to 1,6 glycosidic bonds. And so then you also need to have other enzymes apart from the alpha and the beta amylase, which we just talked about. You also will need enzymes that can break the glycosidic bonds at the branch chains, the alpha 1,6 glycosidic bonds. And so you have the enzyme glucoamylase, which is also able to break down such glycosidic bond, the 1,6 glycosidic bonds, because you do have also structures like dextrins that are present in a mixture of say if you take such a plant biomass from which you are producing the beer from, you have dextrins also as some of the carbohydrates that are present in there, apart from starch that you are breaking down the alpha and the beta glycosidic bonds in. And particularly you have what is referred to as high gravity beers, or say the process of high gravity brewing. Normal beers generally contain alcohol content of about, say, 5%, thereabout. But you do have beers that are produced to have higher alcohol content, about maybe 7%, 9%, in some cases 10%, 12% beers. What it means is that those beers contain more alcohol than normal. And in order for such, production of high alcohol beers, so the high alcohol beers are what are being referred to as the high gravity beers, and the process is being referred to as the high gravity brewing. So that in order to have more alcohol being produced, then you need to have, the breakdown of more carbohydrates into, the more polysaccharides into simple sugars that will get fermented. And so that is how you can have also, this glucoamylase being applied in addition to the alpha and beta amylases. Hilda says the alcohol, is it the raw one? I don't know what you mean by raw alcohol. Maybe you want to explain that to me. Well, what do you mean by raw alcohol? I don't understand it. So clarify what you mean by raw alcohol, and then I can respond to your question. The one that is not mixed. Mixed with what? I still don't get it. So if you're talking about perhaps what you mean to say is those who do fermentation of alcohol just into ethanol, that maybe the percentage of the ethanol is very high, not that is almost like they say an absolute ethanol. Maybe that is what you are talking about. Yes, that is produced by fermentation processes as well. Ethanol produced by fermentation processes as well that way. But in beers, for instance, the production is not just separation of such an alcohol, because it's not distillation. So you have to distinguish between this process of brewing from distillation. Distillation will give you such high, purity of alcohol, but what you have to understand is that, for instance, in the local process of distillation, when they take, say, palm wine, or they take maize or anything to do some fermentation from it, you see that after they have the fermented products, then they now do distillation. That is to separate the ethanol from the water content in it. So yes, you are saying unprocessed fermented drinks. So you see, you say unprocessed. So if you have unprocessed fermented drink, you know that there is high content of water also in it there, together with the alcohol. But in order to have what you did refer to as a raw alcohol is, I think, so the raw, maybe the raw you mean, but if you say the raw, you say it's not mixed. If you say it's not mixed, then it means you are referring to the alcohol which is produced by distillation. So after you have a raw unfermented drink, then you can produce high, purity alcohol, high purity ethanol from that unprocessed fermented drink by distillation. It's a form of separation of the ethanol from the water content, because the boiling point of ethanol is lower than that of water. I think it's about 70 degrees Celsius, and you have water at 100 degrees Celsius. So when you apply heat, then you have the alcohol, getting vaporized. And then once you condense it, you then have a pure alcohol that is being collected, and water is left behind. So, I think you should have this understanding that to produce such high purity alcohol, you need to use, process of distillation to do such a separation. All right. So I hope Hilda gets that. Now, we can look at also cellulase. Cellulase. So you see, you also have, you remember we talked about the fact that you have the woody part of plants containing cellulose, which is, also a major component in indigestible, parts of plants, which means that, apart from the use of this alpha, beta amylase together with what we just saw, glucoamylase, there can also be cellulase also in there. And so this cellulose, there can also be cellulose in there. So the cellulose also needs to be broken down. If you want to increase the yield of production, then you have to take into consideration all these different carbohydrates, so that the enzyme cellulase is also part of this mixture of enzymes that are introduced into the production mixture, into the bioreactor. And this goes to hydrolyze cellulose, also to simple sugars, say glucose. And then such glucose can then be acted upon by, say, yeast, say the microorganism, which is introduced to carry out the fermentation process on the sugars. Lipases are also used here again, also to break down fats into free fatty acids. So here too, you want to also think of the fact that, see, when we were looking at factors that affect enzyme activity, we did look at concentration of enzymes, concentration of substrates, in addition to, say, temperature, pH, all of that being controlled. And here you need to take note of the fact that the amount of enzyme, say the lipases, which is being introduced here, also has to be optimized such that it doesn't result into the breakdown of shorter chains, shorter hydrocarbon chains, which will be resulting into what we refer to as rancidity, because then you have these short chains that can get oxidized and be producing some aldehydes, which will also be causing some off tastes in the beers. So of course, you have this the use of lipases to reduce the off flavors, which can be caused by some of these oxidation processes. So at the end, there will be some cleaner taste in the final beer, which is being produced. Now, we move to the textile industry. The textile industry. So in the textile industry, of course, here too, the things we've talked about, like cellulose, it's actually a major component of fabrics, because that's the indigestible part of plants. What do we use in making these fabrics? Cotton, wool, silk. All of these are coming from plant sources, which consists of, cellulose. And so you have the process of, biopolishing, what is referred to as biopolishing, which is the use of cellulase that, hydrolyzes some of the cellulose fibers. So if you take notes, the diagram at the top is showing, three different fabrics that, at very close range. With some microscopy, you can see that you have some little hairs that are at the surface of these fabrics. But so that is before the biopolishing. But when you look below, that is the diagram after biopolishing by enzymes. That is after the application of enzymes to the fabric to hydrolyze these, small cellulose hairs that are protruding from the fabric. You see the fabric is looking finer. It's looking smoother because those little hairs are removed from there. And this is to reduce, things like peeling. Like you have a fabric that when you are wearing the clothes, depending on maybe the parts of the cloth that touch surfaces and things like that, you begin to see that some places are wearing off by forming some little hairy balls and making the whole clothing not looking nice at those particular positions. And also you have biopolishing, causing enhancement of the fabric. The fabric is softer, the fabric is smoother and shinier. That is, the luster is improved for the fabric. I mean, previously there has been a use of different chemicals, some chemical substances which are used for such polishing processes. But with the advent of sustainability, sustainability, green chemistry, things like that, then the use of enzymes to do this, has become a popular thing because this is seen to be more eco-friendly, more sustainable than the use of, say, some harsh chemicals to do this. So that is bioprocessing, that you see that cellulase is being used to remove these small hairs, what are being referred to as microfibrils. We also see the use of pectinase, the enzyme pectinase in industry, which breaks down another polysaccharide, pectin. Now what you see is on the right hand side, you can see at the top, that is wool. Wool, before the process of scoring. So usually for wool, yes, the term scoring is used. That is in the textile industry. If you go back, you see the use of terms like boiling out, which usually is used when it is for the treatment of cotton. And then you may also hear terms like boiling off, which is typically used in industry for fabrics like silk. But they refer to the same process. In the past, a lot of these had been done with high temperature. I mean, that is why you hear terms like boiling out or boiling off. But with the use of enzymes, this same process, scoring can be done at lower temperatures. And of course, that is also more eco-friendly. And so you see the picture below here, that is in a textile industry where you have wool, which is soaked with some enzymes, a pectinase in there. And you see that wool on the right-hand side, the wool before scoring, you can see is a lot of dirt in there. But after scoring, with the picture below, you see that the wool has become finer. A lot of the dirt has been removed from it, and such dirt is coming from pectins. Like these are like some there are some polysaccharides which sort of form like dirt. There are impurities which you find also in cotton fibers, you know. So if they are not removed, it also does not help with say the uptake of dyes by the fabric when the fabric is being produced. If you introduce dyes because of these deaths, the fabric is not able to uptake the dyes very well. And so yes, one of the processes in the textile industry for the production of high quality fabric is scoring, in which you have the use of pectinase for the removal of these pectins. Another enzyme is lactase. So lactase oxidizes phenolic compounds, and it's an enzyme which is used in the process of biobleaching. This is usually in the production of denim. So denim is what we call jeans, the fabric which is used for jeans is denim, and in the processing of denim, we have a stage we refer to as biobleaching, where also, yes, in the past some harsh chemicals were used for such purpose, and this is to remove phenolic compounds. Plants contain a lot of phenols in them, and many of these phenols do have some brownish colors, that's sort of make the fabric look not so clean. And so you do have treatment of the fabric in the production. You do have this process of biobleaching. And so you can do a comparison. The pictures I have brought below here, you have untreated denim, and you see it's not so fine. In the middle, you have a lactase treated denim, and you see it's the fabric is much finer than what is untreated. And compare that, the lactase treated denim with the chemically treated denim. And this is some research that has been done to compare lactase treated and chemically treated denim, and you see that the chemically treated denim has some destruction of the color of the denim. You see that the lactase treated denim looks much better than the chemically treated denim. You do see this kind of biobleaching being used in the production of different types of jeans. You see the jeans that looks sort of more kind of whitish, different blue colors, deep even some having some kind of stripes, things like that, treated with, they have some snow treated and different kinds of jeans. You've been seeing this on the market, at different kinds of jeans. And such different kinds of jeans are produced from this process of biobleaching. In fact, I remember sometime past, there were even some people who were doing some locally design jeans. They get these, particularly they get some white jeans and begin to give it some kind of dye. Of course, those ones are not being making use of any enzyme in there. They're just using some different dyes. But if you went to some, if you go to, sometime in the 80s thereabout, uh, where some of these uh, people in music, like uh, rock music in Europe, America, you see, they do produce some locally made, no self-made jeans. They get the material and then they make use of some chemicals and produce some kind of very different kind of jeans types. Because those jeans actually had been a fabric which was referred to as um, workers' clothing, because that was, the hard material that was being used, was being worn by people working in industries and different places. Uh, and so it has been a Western clothing in the past for such. Of course, maybe if you went to a place like United Kingdom, they were mostly wearing khakis. So jeans is more of American, and you have the British. They were using more of khaki, which is why I think Ghana too, somehow we got introduced to khakis first before jeans came. And so you see that even in our schools, senior secondary schools especially, even junior secondary schools that at the time had some boarding houses and things like that, you realize that there was that introduction of the use of khakis in these schools. Nowadays, you have even primary school students making use of jeans as a Friday wear, for instance. So you see, these are some trends, changes in things in a society. So, well, lactase, yes, that's an enzyme which is used for oxidizing phenolic compounds in textile industry. Now, the detergent industry. The detergent industry. Again, lipase. So you see, similar enzyme which was being used in the food industry, in the textile industry, and they can be applied in different ways. So that lipase, knowing that lipase can break down fats and oils, when it is integrated into detergents, the lipases can then help to break down oil-based stains or some greasy stains in clothes, because these are oils that can be broken down by the enzyme. And so you see, you don't necessarily have to, usually what you do when you want to remove stain, which is oil-based, you boil the fabric, or you use maybe hot water, boiled water, or even if you're using a washing machine, for instance, you set the temperature to say maybe 60 degrees Celsius, and then so that you make use of the heat to melt out the grease out of the clothing. But with the use of lipase in the detergents, which you are using to remove the stain, then there will not be the need to use such high temperatures for the removal of the stain. So that's an advantage to that. That's a more eco-friendly way of removing the stains from the clothes. So you have lipase doing that. Now, it is not every stain which is oil-based. You have stains of different forms coming from different compounds. So you have some carbohydrates which can also be stained. I mean, we just saw even pectin, because we saw pectinase, which was being used in biopolishing, in the process of scoring to remove those stains, which were pectin based. You also do have mannan based stains, and the mannan is what you see also below. It's a polysaccharide, just like you have the polysaccharides, say starch, glucose, molecules that are bonded together with glycosidic bonds. You do here have some, let's say, a unit of mannan containing 4 glucose molecules, and also having some galactose bound, the galactose being the blue, which is bound on that second glucose residue as a mannan structure. So what it means is that mannan is going to be, repeated units of these. Will give you, the full structure of mannan. And these also have, they also sort of become some stains in what is referred to as, what you call gum. So, you see plants have some gums that are produced in them, like in beans. You do have these gums. You have ice cream having some gum in it, and even some of these sauces which are used. You have gums in them. I mean, you can think of it in a very simple way, like when sometimes mistakenly somebody places some gum somewhere and it touches your clothing, how it stains the place. Particularly maybe some chewing gum or something. You try to remove it, and some of it remains in the fabric. So you can relate with it that yes, gum can be some stain, can form some stain for fabrics. And those gums contain a lot of this polysaccharide, mannan. And so the enzyme mannanase, when it is integrated into fabrics, helps to remove those stains which are mannan based. So yes, that's another enzyme which is used this time around in the detergent industry, and specifically it's for the removal of mannan based stains, which are gums that become stains in fabrics, can be removed by this. Again, pectinase, which we saw in the textile industry, here again being used in the detergent industry. So yes, just the same way as is being used in, say, the process of scoring the production of these fabrics, the same way you see pectinase hydrolyzes pectin. And so you see pectin is also such a polysaccharide. You're seeing a unit of pectin from 5 units of glucose together. And so all what it means is you need pectinase to break down this polysaccharide into simple sugars. And so those simple sugars can easily, get soluble in water. So when you are washing with a detergent and that detergent contains pectinase, and that's, fabric that you are washing is stained with some pectin-based stain, then pectinase breaks down this. So if it breaks it down into glucose molecules, these glucose molecules easily dissolve in water, and that's it. The stain is removed. And And a lot of it will be, a lot of such pectin-based things come from fruits and vegetables, particularly from, say, maybe things like orange peels, for instance, um, can stain clothes. And from them, you do have this pectin, which is a major component of the stains that come from such. So yes, that's pectinase also being used in that sense. Now, in plants, you also do have lots of phenols and phenolic compounds. So phenol and its derivatives like the oxidized, the oxidation compounds from phenols. So you see phenols, as benzene ring with a hydroxyl group on it. You can have those hydroxyl groups getting oxidized to a cyclic ketone group to have a quinone, and you can have multiple quinone, this say 1,4-benzoquinone, 1,2-benzoquinone, 9,10 anthraquinone. You have a lot of these quinones. There are many, so many of them. And in fact, many of these phenolic compounds, especially these quinones, are responsible for the flavors that you observe in things like wine, tea, coffee, cocoa. See, they have some specific flavors in them, and these flavors are actually coming from these phenolic compounds. And these phenolic compounds, they sort of have some brownish color. They are the formation of these oxidation compounds from these phenols actually is what results into, say, what we call a food browning. The browning in food, that maybe if you cut maybe banana, if you peel banana, or even if you put a banana down for some time, you see it turning brown. Or maybe you cut apple and you see the apple is getting brown and things like that. So you have these phenolic compounds that when they get exposed to oxygen, they begin to get oxidized, and then they form such brown colors. The same way in the processing of things like cocoa, wine, tea, coffee. You have a lot of these phenolic compounds in them, and they can also be a source of stain, because if you have wine containing a lot of these phenols and phenolic compounds, tea containing a lot, coffee containing a lot, cocoa drink, and you have it staining clothing, then you can make use of, say, the enzyme lactase, which easily oxidizes these phenolic compounds, breaks them down, especially the complex phenolic structures, breaks it down into smaller compounds, which can easily be removed from the fabric. So yes, lactase is also an enzyme which is used in the detergent industry. Now, in a biofuel industry, this is another area where you want to have enhanced production of bioethanol. Usually, these biofuels are bioethanols that are produced from biomass. And I think the country that produces most of this bioethanol is Brazil, and they produce it from sugarcane. They grow a lot of sugarcane, and then they use a combined harvester, harvest the sugarcane. That's a biomass containing a lot of sucrose there, a lot of sugar, a lot of carbohydrates in there, and mash it into a bioreactor, and introduce microorganisms to carry out the fermentation process into ethanol. So lots of ethanol being produced from there. And so you also here, apart from the fact that you are using a mixture of enzymes that we have seen before, like alpha amylase, beta amylase, pectinase, all of that, cellulase, so that you can have the breakdown of the different polysaccharides into the simple sugars for them to then get fermented to produce ethanol, so that you don't just have some of the carbohydrate broken and a lot of it will go to waste. You also do have the enzyme phytase. So phytase breaks down phytic acid or phytate. Phytic acid or phytate is this say indigestible form of phosphates. So because as you can see, you have lots of these phosphate groups that are bound onto this. You can say it's a derivative of monosaccharide, which has, so it's a monosaccharide which has been modified such that phosphate groups have come to replace all the hydroxyl groups which were on these carbons before. And you need phytase that will break off all of these phosphate groups, which then can be used by the microorganisms that are being used, for the fermentation process. Because these phosphate groups, you remember that the phosphates, activate biological processes. So yes, when they are broken down, those phosphates, they can even go into production of, say, ATP, which will serve as cellular energy for the microorganisms. And so yes, phytase is one of those enzymes which is used in biofuel production, and that actually enhances the efficiency of the biofuel production. If that's not introduced, all of these phytase will be going to waste because it cannot be broken down by amylase. It will not be broken down by either alpha or beta or the cellulase or pectin. So this specific enzyme will also be in that mixture of enzymes that are being used in the production of the biofuel. Right. I think maybe this should be the last example we take. Also under biofuel production. This is the beta-glucosidase. And the beta-glucosidase is responsible for breaking down cellobiose. So when we say cellobiose, what we mean is, say, a disaccharide, which is formed from cellulose. The cellulose is a larger molecule that undergoes some breakdown. Say, under the activity of cellulase, you have this molecule being broken down. And when it's being broken down, you do have some disaccharides formed out of it. And these disaccharides, which are reducing sugars, that is to say that you have the sugar, the disaccharide with one of the anomeric carbons being opened, and so because that anomeric carbon is reactive and can undergo further reaction with any other molecule there to form a glycosidic bond. You have this beta glucosidase that acts on these disaccharides from cellulose, which we are referring to as cellobiose, breaking them down into glucose molecules. And so once they are broken down into those simple sugars, glucose molecules, then you can have those glucose molecules going into the process of fermentation that and that is to produce the ethanol, the bioethanol, which is being produced as a biofuel. So it's a way of also increasing the efficiency of the production of the biofuel. So you can see it's not just a single enzyme which is being used in the production of these things, but it's a mixture of these enzymes. Put all of these enzymes together to make sure that all the possible carbohydrates which can be present in a mixture will have some enzyme which will be responsible for breaking them down. By that way, you will be able to have the um, the yield of your product increased. All right. There's one more. Yes, you have when you take hardwood. See, we've been talking about the production of these things from hardwood. Again, in that biomass mixture that is being introduced into the bioreactor for the production of biofuel, you do have. So I think we'll come to this course has biomembranes as part of it. Even before we come to that, at least we can take a look at this to see that you have a cell membrane. Let's say it's a plant cell membrane, which is the lower layer there, and you have a protection of the cell wall of the cell membrane of plants, which is a cell wall being made up of, you see, a network of pectin, cellulose fibril, hemicellulose. There are also some soluble proteins in there. Before you have, some also covering middle lamella, which also contains lots of pectin in there. So you see, it's a network of different carbohydrates, which we just looked at. Pectin, cellulose, hemicellulose, all of these, forming this complex, which is the cell wall, giving protection to the plant cell. Now, in that production of biofuels, hemicellulase is used for the breakdown of hemicellulose, which is, as you can see, is a major component of the plant biomass. So that when it's broken down, then of course into simple sugars, it will then become fermentable. Those sugars are fermentable. So hemicellulose is broken down into simple sugars like xylose, arabinose, and those can then go into the fermentation process. Obviously, if you break down this hemicellulose, then of course this whole network, which also contains pectin and cellulose, all of it will also be exposed to the breakdown of the various enzymes that we just looked at. All right, so I think this is where I'll end it and open the floor. If you have anything, you can write in the chat. If you want to say anything, you can raise your hand, and I will enable your microphone. You speak. I will respond to you. The floor is open.